next speaker's up, Kailey, Kowalski.
So, hi, my name is student Dr. Kailey Kowalski. I worked with Dr. Zaidi over the summer and with the help of Dr. Sarzor in the lab. The title of my project is Effects of Hypoxia and Reperfusion on a Neuronal Calcium Transporter. This is just a quick outline of what I'll be going over. So stroke is a major health event in the United States. It is the 5th greatest cause of death and a leading cause of disability. There's 2 types of stroke, ischemic and hemorrhagic. My project focuses on the concept of ischemic stroke, which is when blood flow to the brain is blocked, resulting in hypoxia. The hypoxia created during ischemic stroke causes a decline in oxygen ATP, an elevation of intracellular calcium, generation of reactive oxygen species, membrane damage, and ultimately neuronal cell death. The therapeutic approach to stroke involves restoring blood flow to the brain, which further increases reactive oxygen species, causing more tissue damage and more cell death. So while many ATPases have been studied regarding ischemic reperfusion injury, the work done in the plasma membrane calcium ATPase, or PMCA for short, is pretty minimal. PMCA is a high affinity calcium transporter that uses ATP to pump calcium out of its neurons, and it's critical in its ability to restore calcium homeostasis and maintain neuronal functioning. So we have found that PMCA is highly sensitive to oxidative stress, and when put in a high oxidative stress environment, it undergoes inactivation, aggregation, and fragmentation. Because hypoxia causes a reduction in ATP, we hypothesize that PMCA activity will decrease under hypoxic conditions. And because reperfusion causes an increase in oxidative stress, we hypothesize that PMCA activity and its protein levels will further decrease reperfusion. Just some clinical significance. Interventions to preserve PMCA and boost its activity may be developed for future therapeutic approaches to patients with ischemic stroke. Some examples of this might be vitamin E, which can act as an antioxidant, and polyunsaturated fatty acids. So I first approached my hypothesis by using SHSY5Y cells, which are human neuroblastoma cells. I differentiated the cells for 5 to 6 days in 10 micromolar retinoic acid. And as you can see, the cells from like the red arrows, you can see that the cells become more neuronal and develop connections with one another. After differentiation, the cells were subjected to hypoxic conditions for various times. And then to create a reperfusion environment, I then reintroduced the cells to 21% oxygen. PMC activity was determined by measuring the amount of ATP hydrolysis at all the time points. Protein levels were measured by Western blot, and total reactive oxygen species by flow cytometry. So focusing on the results, this first figure shows the results of hypoxia and PMC activity. There's 3 time points shown. There's 2 hours, 4 hours, and 6 hours. The blue bars represent the control, and the green bars represent the hypoxic conditions. And as you can see, for all time points, there's about a 50% decline in PMC activity, including at the 2-hour mark, which is the lowest hypoxic time I've done. Although this is expected though, because there's a decline in ATP with hypoxic conditions, and PMCA relies on ATP. So this next graph can be like a little confusing, so just bear with me. It focuses on PMC activity with hypoxia followed by reperfusion. The blue bar is the control, and the green bar is 2 hours of hypoxia, 4 hours of hypoxia, 6 hours of hypoxia, as seen in like the previous slide. So the next 3 bars represent the reperfusion conditions. Each gray bar is 30 minutes of reperfusion, the yellow bar is 1 hour of reperfusion, and the orange bar is 2 hours of reperfusion. For each condition, after 30 minutes of reperfusion, there's an initial increase in PMC activity for all conditions. And this is possibly due to the reintroduction of oxygen and the synthesis of ATP. Following 1 hour and 2 hours of reperfusion, there's a greater drop in PMC activity, with the greatest decline in PMC activity being at the 6 hours of hypoxia and reperfusion, as compared to the lower hypoxic times. This decrease in PMC activity could represent the permanent inactivation of the ATPase because of the increase in oxidative stress. So focusing on oxidative stress, we know that it increases because total reactive oxygen species was measured under 2 hours of hypoxia and various reperfusion times. And as you can see, there's a sharp increase in total reactive oxygen species following reperfusion. This last figure is looking at PMCA protein levels and focusing on 2 hours of reperfusion for all conditions. With 6 hours of hypoxia and 2 hours of reperfusion, which is seen in this box over here on the right, there's about a 40% decline in PMCA protein levels. And as I stated before, PMCA undergoes aggregation, fragmentation, and activation in high oxidative stress environments, which could account for this decline in PMCA protein levels. So in conclusion, PMCA activity decreases with hypoxia and reperfusion. PMCA protein levels are also reduced in reperfusion, and PMCA activity correlates with an increase in reactive oxygen species. So in the future, I hope to measure the intracellular calcium levels during like hypoxic and reperfusion conditions, determine cell viability, and determine protection of PMCA by antioxidant treatment, protease inhibitors, and membrane stabilizing agents. I wanted to thank my mentor, Dr. Zaidi, for helping me out over the summer, Dr. Sarzar for also helping me a bunch, and Kansas City University for allowing me to conduct my research, as well as the rest of the lab. So thank you.
We have time for a 1-minute question.
Yeah, I was just wondering if you said one of the things is thinking of potential therapies that might increase this PCMA activity. Was that something you were alluding to, or? Yes.
So I wanted to like include like an antioxidant, which will reduce the oxidative stress. Unfortunately, the experiments take a significant amount of time, so I have not had the time to do that. But hopefully I can pass that off to like another medical student. And I know some people in our lab are working on that currently. Like I mentioned membrane stabilizing agents. And I know like currently somebody in our lab is doing that. So it's in the works, which like working on like DHA, which is, I mean, fish oil. So if you could preserve it with something that simple, that would be really great for therapies.
There you go. That's some Dr. Zaidi influence in there with the fish oil. I know that.
Yeah.
So great. And listen, I would just throw out to the group, one of the things is, as we talk about research, especially at KCU, think longitudinally. Think of passing your research on to students behind you. You will benefit from it. It's the team piece, right? They'll continue the research. You'll increase your productivity. You'll increase your publications. And there's in the longitudinal opportunities there by continuing the research over time is an advantage for everybody. So just, I love the idea that we've talked about building that pipeline of research and continuing those threads of research questions over time.
I like that too. I like, you know, the anatomy.
Dr. O'Connor, I prefer fried fish or fish oil, but just a little loop. So one of my research track students will present her data tomorrow on the DHA and the effects on PMC, and it certainly boosts the activity. Go fish.
Go fish. There you go. Thanks.
Thank you very much.